Hello, welcome back. In this video, we will cover the acid-fast staining principle and procedure. Acid-fast staining is used to identify some specific bacteria that do not stain well with gram stain. Clinically, the most important application of this stain is to detect Mycobacterium tuberculosis in sputum samples to confirm or rule out a diagnosis of tuberculosis in patients. Before we move on to the procedure of acid-fast staining, we must know the different acid-fast cells that can be found in clinical practice. A mnemonic for acid-fast cells is LMNOPQRST, L for Legionella, M for Mycobacteria species, N for Nocardia species, O for bacterial spores, P for cyst of protozoa, Q is vacant, R for Rhodococcus, S for sperm head, and T for Taenia saginata eggs. AFB staining has an interesting history. In 1880s, tuberculosis was responsible for 1 out of 7 deaths around the world. It was called white plague or consumption. The agent responsible for tuberculosis was not discovered by then. In 1882, Robert Koch reported the discovery of tubercle bacillus in a patient sample using a complex staining procedure. In the Koch's procedure, the heat-fixed smears were placed in an alcoholic solution of methylene blue. It took prolonged immersion in dye for up to 24 hours for the bacteria to get stained. Robert Koch discovered the hot staining method and found that when immersed in methylene blue dye at 40 degrees centigrade in a water bath, it took only 1 hour for the bacteria to get stained. He used Vesovin dye as the secondary dye for 1 to 2 minutes. Mycobacteria appeared blue while all other bacteria appeared brown after this staining technique. Koch's procedure was not very effective for visualization of the mycobacteria. It was modified in the same year 1882 by Paul Ehrlich, who replaced methylene blue with basic fuchsin, added aniline as a mordant, and he was also the founder of the acid-fast property of the mycobacteria. The procedure was further modified by Franz Ziehl, who used carbolic acid in place of aniline as the mordant. Ziehl Neelsen combined the basic fuchsin and carbolic acid to make a single primary dye called carbolfuchsin. This method is now popularly known as Ziehl Neelsen staining or Zn staining in short, and is widely used for staining of acid-fast organisms. Zn staining uses acid as the decolorizer, and therefore the organisms that stain with this are called acid-fast organisms. Other than the hot staining method, that is Zn staining, there are 2 other common acid-fast staining methods that do not use heat. These are Quignon method and auramine rhodamine fluorochrome truant method. Let us see the similarities and differences between gram-positive, gram-negative and acid-fast bacteria. The cytoplasmic membrane of all 3 bacteria is covered by a peptidoglycan layer that is equally thick in gram-positive and mycobacteria but thin in gram-negative bacteria. The gram-negative bacteria have a thin outer membrane, but the mycobacteria have an outer membrane that is a thick layer of arabinogalactans and mycolic acids. The mycolic acids resist staining with the common staining procedures like gram stain. The use of heat during staining with the primary dye, also called the hot method, helps to drive the primary stain into the waxy cell walls of mycobacteria. Now we move on to the principle and procedure of Ziehl Neelsen staining. Staining of acid-fast cell requires higher concentration of primary dye and a heating period. However, once a stain is introduced into the cell wall, removing it with an acid-based decolorizer is even more difficult. Hence, they are called acid-fast organisms and will appear red using bright field microscopy. We add a secondary dye to stain the decolorized non-acid-fast cells and field debris that will appear blue. The requirements for Zn staining include glass slides, wax pencil, spirit lamp, nichrome loop, distilled water, light microscope, and patient sputum sample. The reagents used for Zn staining are The primary stain, carbofluoxin, also called the Ziehl Neelsen solution, decolorizing agent, that is 1% acid alcohol, and the secondary stain or the counter stain, which is 1% methylene blue. Procedure for Zn staining. First, we take a clean, dust and grease free slide and make a small circle in the center of the slide with a wax pencil. The smear for suspected tuberculosis sample should ideally be prepared in a level 2 biosafety cabinet after wearing proper personal protective equipment. Now sterilize the nichrome loop and allow it to cool. Smear can be prepared directly from a patient specimen like sputum or after it has been processed by centrifugation to get a concentrated material. Using the sterile microbiological loop, pick some small quantity of sputum and apply it to the center of the slide. Slowly spread the sample to make a thin film. The size of the film should be about 1 cm in diameter. Avoid any actions that would splatter droplets of the sample into the surrounding area. Prior to heat fixing, smears should be allowed to air dry completely within the biological safety cabinet. It may take a few minutes for the slide to completely dry out. Pass the air-dried glass slide 3 to 4 times through the blue part of the flame with smear side facing up. The flame should touch the slide for 1 second each time. Do not overheat the slide as it can lead to charring. Other methods for fixing include on an electric slide warmer at 65 to 75 degrees centigrade for 2 hours or at 80 degrees centigrade for 15 minutes. We can also fix the slide with 10% formalin or with 5% phenol in 70% ethylene oxide for 5 minutes. Heat fixation fixes the smear to the slide. It keeps the morphology of the bacteria intact. It also kills the bacteria, thus making the slide non-infectious. However, there are some cautions to be kept in mind. Flame fixing may aerosolize the organisms from smear. Insufficient heat or time can lead to smear washing off. The bacteria in the dried and fixed smear look colorless and indistinguishable from each other. To begin the staining process, cover the smear with primary dye carbofluoxin for 5 minutes. Simultaneously heat the dye using a spirit lamp. Heat gently to produce steaming but without boiling. Apply heat at intervals to keep the stain hot. Do not allow the staining solution to dry on the slides. Add fresh solution if necessary. Allow the dye to stand for 5 minutes. Drain off the excess dye and wash with water. If you observe under microscope, after this step, all bacteria stain pink and differentiation among various bacteria is not possible. Next step is decolorization with 1% acid alcohol. Flood the slide with acid alcohol. Allow it to stand for 1 minute. After 1 minute, wash with water. Repeat the process till there is no more pink dye in the draining fluid. Repeat decolorization with 1% acid alcohol. Again allow it to stand for 1 minute. After 1 minute, wash with water. Repeat the process till the effluent is clear. If you watch under microscope, the non-acid fast cells get decolorized by the acid alcohol while acid fast cells retain the pink dye. Add secondary dye or a counter stain. Flood the slide with methylene blue and allow it to stay for 30 seconds. After 30 seconds, wash with water. Dry the slide using blotting paper. Once dried, remove the slide from the absorbent paper and then observe under 100x objective of microscope. Let us see how some of the commonly found acid-fast organisms or cells appear under the microscope after acid-fast staining. The method described in this video is mainly used for Mycobacterium tuberculosis and non-tubercular mycobacteria. For other acid-fast cells, we need some modifications in the decolorizing agent. This picture shows pink tubercular bacilli that appears thin with beaded appearance on Zn staining. They may appear singly or in clumps. This picture shows lepra bacilli that are thinner than tubercular bacilli and have non-beaded appearance, may be found singly or in clumps called globi. This is Nocardia species, which shows beaded appearance, and bacilli are elongated and filamentous in shape, and it is also partially acid-fast. These are bacterial spores that can be stained using acid-fast Zn staining technique. So this was all about Ziehl Neelsen staining for acid-fast bacilli. Hope you liked the video. Please like, subscribe, comment and share. Thank you for watching.